It's a lot of fun to come back to CDC. When I finished my first residency in internal medicine, I applied for an EIS position because I wanted to work in infectious diseases, and I was told that we don't give you that option. You'll go wherever you want to go. And every time I would meet Joe McDade, I would have that conversation with him. But we've come full circle, and now most recently we worked with Steve and Terry with AFM, but Stuart and I have done a lot of work together over the years with interesting pathogens. Sharif and I have done work going all the way back to West Nile in '99, and Steve Monroe and I did a lot of work in Minnesota on the pork worker inflammatory neuropathy. So there's been a lot of intersection between our laboratories and our efforts over a period of years. These are all the things we study now. It's no longer just viruses, but also bacteria and fungi. And although Jon Snow is cool in the UK, he's been supplanted by Jon Snow in Game of Thrones, as I was reminded, though I have yet to see Game of Thrones, but there's also apparently a Jon Snow there. So this is obviously from the 1850s, the cholera outbreak, and we see Jon Snow breaking off the pump handle, which everybody said, you know, people initially at first blush say this is what arrested that outbreak, when of course that wasn't true, but it makes for a great picture. And now, of course, what we do is completely different. We look for emerging infections based on analyses of hotspot maps, and we bring computers and sequencers to the table to try to understand this. I really began my career in infectious diseases when I was working as a neurology resident at UCSF, and Julie Gerben and I both staffed Ward 86, as it was then known, which is where we took care of patients who had, at least in my domain, they either had CNS lymphoma or they had Kaposi sarcoma. And because it took 2 years to recognize the agent for what it was, what I decided to do was to go work with Michael Oldstone and Floyd Bloom at Scripps to try to understand ways in which genetic methods can be brought to bear. This paper came out in 1985 describing what was a very exciting idea, the idea that there might be one virus that was at that point not yet characterized that was responsible for a major psychiatric disorder. So I devoted several years to trying to identify this and developing the tools that would be required, including in situ hybridization and certain types of interesting RNA blots and in vitro translations and IPs and all kinds of things that no one does anymore. But ultimately, we found those agents, and that then allowed us to recognize the first agent that was identified using purely molecular methods. Then everything pretty much stayed stable until the discovery of PCR, and we came up with various modifications for that and arrays and so forth. And the next revolution came about with Roche 454 in the early 2000s, and we did some very interesting work with Stuart on Bugnic-Buyu and Lujo and other agents, and then Ion Torrent and Illumina, of course now Oxford Nanopore. So in our center alone, we've identified at least 1,500 viruses. We really don't count them as much as we used to do. Probably 5% of them prove to have some sort of impact economically or clinically. And the rest of them are just interesting in terms of filling out the catalog. Now, one of the things that we've been trying to do is to find ways in which we can reduce the cost of doing this sort of work. And the approach that we've taken is to focus on sample prep. And sample prep is not something people talk about. It's not very sexy, but it really does work. So if you use subtraction, which is what I started with, you can get a 10-fold enrichment at best. If you use positive selection, like people did for exon sequencing and so forth, you can get orders of magnitude improvement. This is a system that's really very straightforward. We reported a few years ago that's now licensed, in which you take oligonucleotides that cover the genome of all known vertebrate viruses. You make your libraries, you pull down your relevant sequences, you get the coding sequences of all known viruses. They can vary up to 40% at any point along their genome, and we still recover them. And this reduces your cost for sequencing dramatically. It also gives you a dramatic increase in your sensitivity. So it's actually now equivalent to real-time PCR. So there are a wide range of viruses that have been characterized in this way. We've done this in stool, we've done it in blood, we've done it in lung, and so forth. And it proves to be very, very powerful. So the other advantage to it is that the bioinformatic complexity is dramatically reduced. So instead of having to look at 200 million sequences, you can look at 2 million and get the same sort of data. What you miss are the termini, because we focused on coding sequences because of the limitations of the printing technology. Nonetheless, it's a wonderful system, and you can see here, which I don't have a pointer, so I can't really show you very well, but you can see, for example, the number of viral reads and genome coverage with unbiased high-throughput sequencing done in Geneva versus VIRCAP-seq. And we generated the same kind of data more rapidly at a much, much lower cost. We recently reported the same sort of an approach for bacteria. So you can now characterize bacteria phylogenetically, again with the same sorts of improvements in sensitivity and reductions in cost. You can do something which is even more interesting, I think, and that is you can put bacteria into culture. You can pull out those cultures at 90 minutes or 120 minutes. You can characterize the transcriptional profiles, and you can find biomarkers indicative not only the presence of an AMR element, but function of that AMR element. So this has been very, very powerful, and this is an example that shows you the kinds of variations you see with AMR sensitive and resistant Staph aureus in the presence or absence of antibiotics. This is another example of the same sort of work. Once you identify these, you can put them into other sorts of systems, whether it's nanostring or PCR or whatever you wish. Now, are there instances where we miss agents? Not yet in humans or in mammals. However, when you start moving into other life forms like fish, it becomes a problem. So several years ago, when we were still using only unbiased high-throughput sequencing, we found a novel reovirus that was very important in killing salmon. And in fact, this has resulted in vaccines that have restored salmon stocks. We did similar sort of work with tilapia. Initially, we applied the positive selection methods, and they failed. So we then went back to unbiased high-throughput sequencing. This is an extremely important virus vis-a-vis food security. All these tilapia farms located worldwide. It kills these animals, destroys their livers, gives them central nervous system damage, and it's been highlighted as an enormous threat by FAO. So on the left side, you can see there the in situ hybridization we did with probes that were generated as a result of that work. You can see the icosahedral sort of capsids down there. And this required us to go back to basics. So who here has actually done a northern blot in the past 10 years? Nobody, maybe one person. So what we found initially were that there were 10 genome segments. They had similarities at their termini to one another across the 10 segments. They ranged from 500 nucleotides to about 1,500 nucleotides. And so we made probes and looked at RNA blots. It took us a long time to set up RNA blots again. And we found correlate transcripts associated with each one of these genes. And that then, coupled with some HPLC and mass spec we did to identify individual proteins, allowed us to recognize this as a bona fide virus. Its closest relationship, and it's quite remote, it's 40% in PB1 to influenza C. So it is a novel orthomyxovirus, and the preliminary data that we and others have suggests that it replicates and transcribes in both the nucleus and the cytoplasm. But because it's way off, from what we do in human disease, we've really not pursued it further. So other people may have better data, particularly in the flu branch. Now I'm going to show you a way in which we've tried to integrate not only these discovery methods for molecular biology, but also for serology. So as many of you know, there are recurrent outbreaks of encephalitis in Uttar Pradesh. It really is an enormously sad situation. It hits the very, very poorest of children. Sumya Swaminathan, who was then director of ICMR 2 years ago, asked us to go and try to apply these methods to characterize it. When we went, this hospital was going up in Gorakhpur to house just these kids with encephalitis. And you can see here what contributes to it. So this is sort of the headwaters of the Ganges. You get flooding. You wind up with these children who are the lowest caste within the Dalits, Musahar, which translates roughly to rat eating. And these are the children who have this problem with encephalitis. When we began looking at the samples that we got, spinal fluids, from the children where no diagnosis had been made, we found some things that were surprising. They missed herpes simplex type 1 that they shouldn't have done and other things like this. But in addition, there were these two bacteria that were important, oriented to Tsugamushi and a novel Rickettsia felis, and these were highly associated with disease as well. We're still trying to publish this work, and it's complicated because any of you who've dealt with ICMR know that it makes your publication problems look fairly trivial. Anyway, so by combining the back-cap-seq that I've just shown you and a serochip, which I've not yet talked about, we're able to resolve a large number of these cases, and this has already had an impact. Now, this is the serochip. It was originally developed by Nimblegen. Nimblegen was purchased by Roche. Roche decided they didn't want to print anymore, so the name of the company is now Nimble Therapeutics. Unfortunately, the price of the arrays has gone up about 30%, and they were expensive to start. But what this allows you to do is printed very high-density linear epitopes across proteomes, human proteomes, vertebrate proteomes, whatever you wish. You can subdivide these arrays into 12 per slide, but if you buy the slide and you put the full complement of, say, 6 million peptides on that slide, it's about $3,500 per sample. So it's not for the faint of heart or the weak of wallet, but you can learn a lot, and for discovery, you can then move downstream and define and refine what it is you're working for. So this is early work that was done by Rafal Tokars, who was trying to develop something for tick-borne diseases. And for those of you who've worked with tick-borne diseases, you know that we have an ELISA followed by a Western blot, and it's time-consuming and extremely expensive. So we printed all of the different tick-borne disease agents of which we knew, as well as some that we'd recovered through discovery in ticks in Long Island and in Connecticut. It was 170,000 peptides. And this allowed us then to define specific epitopes by using peptides that overlap, marching across the proteome of these relevant regions, and refine it down to Borrelias, now down to 5 discriminating epitopes, Borrelias 4, and so forth. And this is work that we did in concert with your colleagues in Fort Collins. You can then move these to even less expensive sorts of assays. Here's an example. This is a pepperprint assay, and this gives you very, very discrete data now for about $50 a sample. So ultimately, I think that much of what we do in serology, which has been largely neglected, is going to be replaced by these kinds of techniques. So you can see here now you get these very, very discrete data with anaplasma using this sort of an approach. Now, several months ago, we began working with Terry, Steve, and others on AFM. I'm not going to talk much about it because you all know more about it than I do, but there are certain criteria which are clinical and radiological. And as you know, we have this, this sort of biannual increase in cases that's been observed now for several seasons. And we initially began by trying to use VirCap-seq-Virt because it's sensitive to try to characterize viruses that might be present there. So Steve, you see, you're still there. Terry's there, but we've lost Niche. Don't tell him, Terry. He'll be very upset. I think I have another picture of Niche someplace. But in any event, we didn't find any nucleic acid evidence of either 68 or 71 or other things. But then when we began looking for immunoreactivity using the approach that I've just described for you, there were specific epitopes that began to come up, which were present in the spinal fluid, and others that were present in the serum, which implicated not only enteroviruses, but because of the high level of resolution you have with these chips, because again, you're stepping one residue at a time across the entire proteome, you can find specific reactivity, in this case, with EVD68. And this is now what we think is the most likely agent implicated in pathogenesis of these disorders. And I say that because although it's buried in the discussion of the paper, it really belongs in the results. And it would be there but for ICMR. So when we look back at all these children in Gorapur who had encephalitis and meningitis, where we found these bacterial pathogens, they are at very high levels of titers and antibodies against EVD68 in serum and or plasma. But when you looked in spinal fluid, there were only 3 who did, and 2 of them had active infections with other enteroviruses. So we feel that that's fairly specific. I'm going to switch gears now and talk a little bit about the microbiome, and I promise, Beth, that this is ultimately leading to ME/CFS. We're just setting the stage for the palette that you bring to it. So the Caprice is an interesting program which is focused on HIV/AIDS in South Africa. An observation was made by the Kareems several years ago that individuals who, during a period of observation over several months, progressed from not being infected to becoming seropositive was that they had high levels of type 1 cytokines in the vaginal fluid. So we began a study where just began characterizing these using 16S as a first approximation to ask whether or not there would be differences in cytokine profiles. And I don't expect you to read that, but clearly the colors are different in the high and low cytokine groups. So we then decided to do whole genome sequencing, metagenomic discovery, and so forth. And this demonstrated for us that there were specific microflora that were associated with increased risk of high levels of cytokines and subsequent seroconversion to HIV. And there were other bacteria that appeared to be protective. So P. bivia was the one that was associated with increased risk, and Shuttleworthy and Megasphere were found the other way. So we're actually using this, and I could show you many other examples where we've begun to use these sorts of approaches to try to understand mechanistically why people might be susceptible to certain kinds of viral infections as a function of their microflora. And here's a mechanism by which P. bivia, which expresses large concentrations of LPS, pulls infectable cells down into the vaginal vault. The last thing I want to show you in this era before I start moving into ME/CFS is some work that we've done in autism, which is another very difficult and thorny sort of a problem. And here, it's an issue really of not only the agent itself, but the genetic background and the timing of the exposure. Now, the proof of this really comes from animal model experiments, where if you expose animals to lipopolysaccharide or poly-IC or influenza virus or whatever, if you infect those animals halfway through gestation, those animals that get the virus are huddled in the corner way over on the left. And if you give the same exposure several days later, the animals are active. So same genetic background, same exposure, different timing, profoundly different effect. So with that in mind, we then began using cohort studies to try to investigate whether or not there were specific types of exposures that might be associated with the increase in risk. This is work that we did with the Norwegian Institute of Public Health. This demonstrated, first of all, that if you had episodes of fever after 12 weeks of gestation, there was a 3-fold increased risk in a subsequent diagnosis of autism. And again, this is all using prospectively collected data. If you have high titers of antibodies that appear in the latter part of pregnancy directed against herpes simplex type 2, there's an increased risk. And if you have flu, there's an increased risk. So all of these things taken together suggest that it's not the agent itself, but rather the immune response. And in animals, we can suppress that immune response and we can save stem cells in the central nervous system by using cuprofen, a non-steroidal anti-inflammatory drug. And although we don't have enough cases to demonstrate this, those individuals who took non-steroidal anti-inflammatory drugs during the early part of pregnancy, who had these fevers, there was no effect of the fever on the risk for ASD. So with that sort of as a sort of an introduction to how great pathogen discovery is, I'm going to briefly talk about all the problems. Some of these will be familiar to you. So we found issues with enteroviruses and ALS. This is one, obviously, with MMR and autism, which is almost impossible to get this paper published, by the way. That's why it's actually in PLOS One. But it was actually cited as one of the most highly regarded papers in the PLOS circuit. And of course, you all know we've got outbreaks in the US and we've got outbreaks globally. And I was in Mumbai last week and I tried to make the case that they needed to be proactive. Then we have bornaviruses and ME/CFS. This is actually how I got into this field. I was invited by Brian Mahi, who many of you will remember, and Jim Dobbins, who some of you may remember, because of these reports in Japan that there were individuals who developed ME/CFS as a result. That then led to a large NIH study which found no association, and Bill Switzer, I don't know where Bill is, I hope he's here. Guess he's not. Oh, there you are. Oh, he's online. Okay, so Bill.
People online have trouble logging in.
Okay, all right. Well, Bill Switzer was a critical partner in this work, and Judy Mikovits was a critical partner in this work. She initially accepted it, but now apparently she doesn't. But in any event, we found no evidence whatsoever of XMRV being implicated, and this was a very expensive, very difficult study to do, but it emphasized the importance of getting this sort of work done in a rational way. Okay, now, for the meat of the talk now, I've shown you all the tools that we can bring to bear on the problem. Let's talk about ME/CFS. Right now, it's a clinical diagnosis, and it is a diagnosis of exclusion. So we make sure that people don't have other intercurrent infections with hepatitis C, they don't have hypothyroidism, they don't have other sorts of problems. And these are data that came from what's now known as the National Academy of Medicine. It is not a small problem. It's something like 2 million people in the US alone, and it's very expensive because these people can't work, as well as requiring various types of support. The causes of the disorder are not well understood. Many patients report what seems like an infectious prodrome with pharyngitis and swollen lymph nodes and feeling feverish and having night sweats and so forth. So initially, when we became involved in this, we figured this is what we're going to do, was a standard sort of a pathogen search. Now this is from the work that we did with Brian and Jim Dobbins, who are not on this paper because for reasons unclear to me, they weren't able to do that at that time. But they really supported this work. It wouldn't have happened otherwise. We didn't find any evidence for bornavirus infection, but at that point I did make one observation that I think in retrospect was prescient. And that was that these individuals, as we studied them, had high levels of antibodies to things like beta-galactosidase, suggesting that they had polyclonal B-cell activation. This is really some of the very first work suggesting that there were immunological abnormalities in these patients. And this work was really done back in 1995. We now have a center for solutions for ME/CFS that's funded by the NIH, though not as well as it should be. The clinicians are shown in yellow: Dan Peterson, Sue Levine, Cindy Bateman, and Jose Montoya has been replaced by Moneghetti. And then we have Oliver Fien for metabolomics. We have John Greeley, who primarily focuses on transcriptomics, Tony Komaroff, who develops applications that we use for tracking patients, and then an admin and Columbia-based core, as shown here. So what I'm going to try to do is to summarize some of the things we've already done. We found altered profiles for various cytokines and chemokines in blood, as well as in spinal fluid. We did these multi-center studies and so forth. This is an example of work that we did looking at cytokine expression over the course of the illness. And what we found was that there were individuals who were caught within 6 months to a year or so of the original diagnosis. They had different cytokine profiles than those individuals who had disease for more than 3 years, suggesting that there might be burnout, I think was the interpretation that we rendered at the time. We really don't have an explanation for it as yet. And there were differences in levels of cytokines and chemokines in spinal fluid, and our control group here was not only normal controls, but of individuals who had multiple sclerosis. And you can see here now that there are various abnormalities which we've explained elsewhere. The center has 3 projects. One is broadly considered the microbiology of ME/CFS, which is essentially characterization of bacteria, viruses, and fungi in everything that we can access: feces, saliva, and blood. And the indirect measure of exposure is going to be increasingly important because we're not really going to find anything, as I'll show you, looking at direct measures based on sequencing. Metabolomic and transcriptomic studies that we're doing with peripheral blood mononuclear cells, and then some translational aims where we look at the impact of somebody standing erect, which is a problem for many of these patients, or doing some sort of an exercise test, which gives them stress. One of the key features of ME/CFS is that these individuals don't recover after exercise. And I'm going to show you some data now which I think will explain how this works. One of the things that we're using, which is going to be extremely powerful, is called true culture. And when we actually began to look at drawing samples across multiple sites, there are differences in levels of transcripts and proteins as a function of how things are preserved. A group at Institut Pasteur came up with a solution for this, where they pull blood directly out of the patient's arm, put it into these culture vials, which can be supplemented with a variety of stimuli. In some instances, they represent bacteria or viruses or fungi, and then at regular intervals, you depress this plunger, collect the supernatant, and then the cells below you can extract and use for various sorts of things. So this illustrates the way in which you can reduce the variability. So these are individuals who are exposed, who had cells who were exposed to lipopolysaccharide, and you can see how much tighter the data are. Again, I don't have a pointer, but if you look at the left there, you can see a lot of spread, and as you use this other system, it becomes very useful. And when you lose that sort of spread, when you get better resolution, you can find individuals who have specific genotypes and phenotypes. So that's the approach. So let's talk about the fecal microbiome. So we've actually now done this twice. We had a pilot study that was funded by the Hutchins Family Foundation, which was 50 cases and 50 controls. And then we had a larger study funded by the NIH, which is roughly twice that. And if we look at the Venn diagram showing what comes up as being common, we find that there are 15 different bacterial types that are highly represented. You can see Ruminococcus, Clostridia species. And why is this important? Well, the Clostridia species are associated with inflammatory bowel disease, mucus degradation, so forth. And then you also have these butyrate producers that are reduced. If you have inflammation of the enteric mucosa, you have the opportunity for trafficking of molecules produced by bacteria into the systemic circulation, where they can then have an impact. So that is one thing that we're now pursuing, and we do that by using metabolomics. The difference is in those individuals who have ME/CFS who have irritable bowel syndrome versus those who don't. Again, you can see the butyrate producers reduced. That appears to be common, but there are other bacteria which appear to be coming up as well. We've looked at the salivary microbiome. There again, you can find differences between cases and controls. So I don't know that these are cause or effect or simply epiphenomenal, but they're at least observations and give you data that you can test in other populations. And I was meeting earlier with Beth and her team, and I think given that you have saliva, we can examine these sorts of issues. You can then ask questions about predicted metabolic pathways that might be disturbed as a function of different populations. And you can see here now that the pyrimidine deoxyribonucleoside salvage, is differentially impacted, and unfortunately, we've lost, a lot of the information on the left side of the screen. But there are inferences that you can make based upon what's known. We recently, and by recently I mean the day before yesterday, completed a proteomics pilot, very, very small, so it's only 40 subjects and 40 controls, but they're a well-matched population. And this finding has been very, very cool, because what we've identified is that there are a number of proteins which appear to be differentially expressed in individuals who have ME/CFS. This is not to say that they all have the same cause, but if you use cancer as a paradigm for understanding this, you have multiple ways in which you can get to cancer: tumor suppressor genes, oncogenes. We think something similar is happening here. So I'll give you an example on the next slide, which supports the notion that at some point there might have been an infection that resulted in an immunological trigger. And these are not bad odd ratios, They're all in excess of 3. Now, what are we actually looking at here? These are proteomic studies that revealed differential expression of immunoglobulin kappa and heavy light chains and heavy chains, which are present at increased concentrations in individuals with ME/CFS. And you can use these immunoglobulin repertoires to try to understand, why is it not moving? Oh, there we go. You can actually use these then to develop biomarker diagnostics which allow you to say this individual is at risk for having ME/CFS. And it's proving to be extremely powerful. We've done similar work with metagenomic analyses of feces and with metabolomics. The metabolomics work we've done in collaboration with Oliver Fien and Dinesh Barupal at UC Davis. They again are focused really on rigor and granularity, so they separately analyze biogenic amines, high and low density lipids, and primary metabolites. Again, comparing this small group of 50 patients and 50 controls with the larger group of NIH patients, we can look for similarities, and here we're finding those as well. So there are specific abnormalities that are referent to mitochondria, to phospholipids, that are associated with ME/CFS and break down differently when you look at individuals who have abnormalities in the bowel and those who don't. So you can see here summarized the glucosylceramide, carnitine, plasmalogens, and so forth. Now, what are plasmalogens? I doubt anybody's heard of them, but they are decreased in ME/CFS. They've been observed to be decreased in a variety of dementias as well, and in Parkinson's disease. So this may be some sort of marker for CNS dysfunction. The last thing I'm going to describe for you in this respect is the Epigenetics Pilot that we've recently done. So we used a system with bisulfite-treated libraries for capturing 5.5 million methylated residues in the human genome. We restricted ourselves to those where we had good data, meaning 20x coverage, and we mapped and annotated their location within specific genes with respect to promoters, non-coding sequences, introns, and so on. And these are the results, and this again, just like the proteomics work I've shown you, is not yet published. You can see that we've got 724 sites in almost 600 genes that met our criteria for hypermethylation. You can see the distribution in the various regions in relationship to those genomes. And this is really very interesting. So ME/CFS is marked there in blue, and you can see that when we look at percentage of methylation, As you begin to look beyond about 60, 65%, you get hypermethylation in the ME/CFS group. And this just shows this data in that way. And it's really, I think this is really some of the most promising data I've seen thus far that has to do with talking about ways in which genomic modification might occur. And there are a number of different gene circuits that have been implicated in this kind of work. It becomes, as you can see, extremely complicated, but many of these are associated with oxidative metabolism as well as with stress. We highlight here the ones where we see the largest changes, and if anybody wants to see these data in more detail, I'll be happy to show you. So I'm just going to demonstrate for you the ways in which we're beginning to tease this apart. When we began looking at the most remarkable findings, at least to our mind, we found that they were related to iron metabolism. Now, if you look at the methylation differences, you know, they're modest, 12%, 11%. I don't know how important that is. We actually then measured RNA abundance, so we created real-time PCR assays so that we could actually look at the products of those methylated genes. And although no one gene is affected in a great fashion, if you sum them all, they could have a profound effect. So this is where we slip into fantasy, and I'll show you what we think is going on. And this is moving into the cell. In this case, we're talking about monocytes and macrophages and somatic tissues rather than red blood cells. So we have our first lesion here, right? We have a lesion at the level of the clathrin-coated pits. We have another lesion as these iron molecules are exiting the endosome. I hate PowerPoint. My new administrator moved me from Keynote to PowerPoint. It doesn't work as well. Anyway, and you can see here now in the mitochondria, there's an effect as it moves through the mitochondria. So in essence, what happens is you have an impact at multiple levels, which should have an impact on your ability to metabolize oxygen, and this could result in weakness and fatigue. And we have to do the same thing with all of these circuits we've identified using all of these different methods. And as we're putting this thing together, it begins to look sort of like an anemia of chronic disease with red blood cell damage and so forth, and oxidative stress. And this is a model that's still in the process of being worked out. I mentioned earlier to you the fact that we're trying to build sera chips. This is our first sera chip, which is designed to look at autoimmunity. But more recently, we've come up with something which is even more expensive but more exciting, I think, and that is the entire human proteome complemented or supplemented by other targets that we think are important. Now, in our experience, 2 to 5% of the patients who come through our research clinics wind up having some evidence of persistent herpes virus infections. Some clinics have more than others. Jose Montoya at Stanford had large numbers of those. Peterson has them as well. So working again with this new company, Nimble Therapeutics, which has replaced Nimblegen, we have these 20,000 odd human sequences. There are 16 mer peptides with 12 amino acid overlap. That's not as good as what we've done in the past, where we have 12 mers with 11 amino acid overlap, but you can see when I tell you these arrays are $3,500 each, you know, we had to make some compromises. We've added peptides that cover mitochondrial proteins. We have control scrambled peptides, which are critical for interpretation of signal, and then we have the reference proteomes of all known herpes viruses because we're concerned that they may be associated with persistent infection, and the peptides that cover the proteomes of all the HERVs, and then because there's frequently is overlap with tick-borne diseases, we've used the refined epitope map that we previously generated that allows us to do this. So we are embarking on a very expensive study that will allow us to examine now that we haven't found anything using direct methods, evidence that at some point these patients were also had an disease. So one of the things that I want to highlight here is that, you know, we had the era of culture and gram stain, and we had sequencing. And I think sequencing is passé. So what's next, you're right, is to examine the history of the immune response so that we can make association between early life infections and later sequelae. And these kinds of chips, which are incredibly expensive, but hopefully it'll come down in cost, will allow us to do this. So in summary of this part, Beth will agree, this is an important disease. It's understudied. It's important economically. It's also important in terms of clinical medicine. All the things that we've done thus far provide biological evidence that this is a bona fide illness with cytokine, metabolomic, proteomic, epigenetic, transcriptomic differences, all consistent with some sort of an abnormal immune response, oxidative stress, and a dysregulated microbiome. So we've come a long way. The high-throughput sequencing thus far has not identified any one infectious agent. So although we'd hoped that we would find something, we've gone home empty there. But the serology will hopefully change that. And the future work is going to have to focus on identifying environmental triggers of pathology in people who are genetically susceptible. And this is going to be very exciting. Now, I couldn't leave without showing some movies. I'm not going to show you the movies, actually, though. But so Contagion, you know. Plague Inc., we help with the game. I did some work on Bond 25, but I can't tell you about it. I can't even tell you if they're still shaking and not stirring the martinis. But it should be cool. I hope it's going to be cool anyway, if they selected one of the things I gave them. And then Utopia, which is about a flu pandemic that's supposed to, you know, wipe out the world except for a select few, maybe up here on Clifton Drive. And then 3 Months, which is the story about an individual who becomes exposed to HIV and worries for 90 days. I don't know. You know, it's not enough yet to pay the bills, but it's fun to work with these people. And then the people who really do the work, who I've tried to identify here. And I'm happy to take questions. Thank you very much. Steve.
So you presented at the end there sort of a model for how some of these injuries might result in effects within the cell. Is it to the point where you could start to test that model by knockouts or things like that in some sort of animal system?
Right. So Steve's question, which is a good one, is can we begin to test some of these models mechanistically using tractable animal models? I think we're not quite there yet. What I'd like to do first is look at the impact of herpes virus infections on different cells in vitro and ask whether or not they're associated with differential methylation of specific genes, for example. And this is something which I think we can do, you know, we can do any time. And then we can begin looking at specific animal models. Maybe as we characterize them phenotypically a bit better, we'll be able to look at like the collaborative cross mice to try to identify genetic homologs of the things that we find in people. Please.
It was fascinating talk, thank you. So in any of the conditions, whether it's ME, CFS, or say the autism study, have any of the clinical cohorts or any protocols in place to start people on anti-inflammatories?
Okay, so
Who may be at risk or who already are not well. And another obvious thing is people with microbiome, the transplants, and so on. So from a therapeutic point of view, where is this leading to?
okay, so I'm just going to repeat questions because otherwise the audience may not be able to hear it. So the question is whether or not, based on the data that I've already shown, people have begun to use some sort of interventions, at least to test them. There are people who are doing fecal transplants, not to my knowledge, with any sort of rigor under any sort of clinical study umbrella, but people are doing it. So those findings are anecdotal. Dan Peterson with a company that makes something called Ampligen, which is essentially polyinosine cytosine, has done some work in an open label fashion which suggests that there was an improvement in the quality of life of individuals who took the Ampligen who had ME/CFS. These data were reviewed by FDA and by people at NIAID and felt not to be compelling. In comparison with data from people who had congestive heart failure, who got various alpha blockers and things like that. So they said, we don't really want to invest further. However, when I looked at those data, although when you put someone onto an exercise bike or tried to put them onto a treadmill, which they typically don't tolerate, there was a very modest, you could even say negligible, improvement in the amount of time they could spend on that stress test. They reported independently that they were able to get out of bed and go to the mailbox and get their mail, which for them, in terms of, wow, that was a great answer, I guess. So what that suggested to me is that their quality of life was improved. And there are people who actually moved to Incline Village to get access to this. There isn't a lot of Ampligen available, and nobody's tested other kinds of poly-IC to see whether or not that would be effective. But in fact, I think that looked very interesting. Now, in terms of autism, there has been no testing yet of that sort. I think that the findings, at least with respect to herpes simplex, would suggest to me that it's not unreasonable to put people on valganciclovir. It's not used by the human TK, and it should be innocuous. And if I were pregnant and I had a history of herpes simplex type 2, I would be on valganciclovir. But I've never been pregnant, so I don't really know. Yeah?
This was very interesting. I was just wondering the abnormalities that you found for patients with CFS. Is the constellation of these abnormalities relatively specific for CFS, or not so much? And this just indicates that it's a bona fide.
So this is a really important question. The question is, what do we know about the specificity of the findings that we have and that will depend on which study we're describing. So the cytokine abnormalities we have in ME/CFS in spinal fluid were tested with a control group that included multiple sclerosis. Why? Because that's what was available. With respect to other things, with stress tests and so forth, we don't have the samples. And one of the problems has been that this disorder does not have resources. So the sampling that we do is always opportunistic. We have to find samples that were collected for some other reason. The budget for these centers is roughly, what would you say, Beth? 15%, 20% of what we were promised? So we don't have the samples to do the appropriate controls. Now for discovery, it doesn't matter very much. So that portion is, yes, they have a disease. And we're hoping that with these findings, people say, yeah, they're not malingering. This is a bona fide illness. And that then we hope will lead to some other additional support. This is the big weakness in this study, is the lack of appropriate controls. Sharif?
Thanks, Ian. Outstanding work and teamwork as usual. Have there been any thought about looking at autopsies or tissues from these patients?
Yeah, so the question is, what about accessing tissues? Because this is an extremely important area. We could perhaps find sites of pathology that might have systemic impact. And the answer is that, you know, I get calls from people who say, I want to donate my body to you. I said, and I always have to say, please don't, because we've had, since I've been doing this work, 2 people who raised funds for us and were engaged in research have killed themselves. And my concern is that we'll actually have people killing themselves to donate their bodies, you know, for this kind of research. So there isn't a lot of tissue. In fact, I'm not any tissue that we could access at this point. And if we do, you will be my first call. So back in 1999, just to sort of toot Sharif's horn a little bit, we were beginning to look at, you know, obviously it wasn't St. Louis encephalitis virus. Sharif had done some work staining with antibodies, broadly reactive flavivirus antibodies. And as a result, what we did then is we designed primers, did the amplifications, separated the products in acrylamide instead of agarose because it gave us better resolution, and we found differences at that point then between the size of the products. And then using chemical sequencing, went back in and found that it was that it was not St. Louis encephalitis virus. Anyway, without Sharif's guidance on that, we were sort of shooting in the dark which panel to use first. We also had probably the worst experience of publishing a paper together with an astrovirus. Do you remember that? So this was a very interesting story. It was a kid with XLA deficiency who lived in Puget Sound near a mink farm. He had astrovirus encephalitis. So we did it was our second 454 outbreak investigation. Oh, it was an outbreak, it was one kid. But in any event, we had 2 sequences out of 40,000 that looked like an astrovirus. So we used those as a hook, right, to sort of pull in the rest of the genome. We sequenced the entire genome tediously in those days. There was no other way to do it, you know, just walking along the chromosome. We then created antibodies, right, to recombinant proteins. Sharif did all the immunohistochemistry, and it took 2 years to get it published in EID. So, just one thing about EID. If you want people to publish in EID, if anybody's involved with EID, let's get an editorial board that really pushes hard, gives us a rapid turnaround. I think 2 years is too long for a publication. Anyway.
So the T is kind of neuron. Do you have any neuroscientists involved in this study?
I'm a neuroscientist. I'm a board-certified neurologist. I don't have any credibility in infectious diseases. Never took an infectious disease fellowship. Was turned down for an EIS fellowship in infectious diseases, sadly, or I would have had a different career. No, I'm very interested in that. And in terms of thinking about this, I think the focus is primarily with ME/CFS on autonomic function and to some extent on cardiovascular work. There's a cardiovascular physician now at Harvard who's doing endocardial biopsies in some of these patients. So they're highly motivated, Sharif, to, you know, if they're willing to have endocardial biopsies. So if you know anybody who specifically is a neuroscientist who wants to work in ME/CFS, I will be delighted to integrate them. Please.
Do you do any crude subdivision of your CFS patients before a study? Because I guess I'm concerned that you probably do have an immune-activated group. There are probably some groups that are slip into CFS through other mechanisms or reasons. So you look, for example, a group that does appear to have an immune trigger.
So the only thing we do prior to inclusion in the group is to test them for certain infections and to test their thyroid function. So we measure T4, T3, TSH, hepatitis C, hepatitis B, HIV, various tick-borne diseases, and those are the only exclusionary criteria that we use. It's very difficult to recruit patients with ME/CFS, period. So, we could let the perfect be the enemy of the good. It's very challenging. It's not like studying HIV. It's very difficult to get these patients. And when you bring them in, it's a day and a half of evaluation. The NIH has an even longer evaluation, I believe. But they're doing things like basing metabolic rates and metabolic chambers and all sorts of things. But it's a smaller number of subjects. Well, thank you very much. Oh, one more.
Just going back to having a sort of a genetic predisposition to environmental triggers, has anyone explored how the immune responses to standard vaccines like flu or hepatitis are, and to give you an idea of kind of historically what may have happened?
So the question is, are there things that we could obtain primarily from historical documents that would tell us whether or not these people had abnormal responses? We don't have that. But what we do have, and that's why I showed you this true culture system, is we have PBMC that have been isolated that are exposed to LPS, poly-IC, staph enterotoxin B, Candida, and there we'll be able to see whether or not they have different responses. Right now we have 40 cases and 40 controls. When we get to 50-50, we will begin that next body of work. And there is a colleague of mine, Daria Unamatz at Jackson Labs, who has found a specific subtype of individuals who have a polarization in their lymphocyte profiles by FACS, suggesting a prior exposure to viruses. So we've now extracted those. We've got very good RIN numbers, up around 9, and we're in the process of trying to figure out what they've seen, because they're going to be studied using this whole proteome chip. Thank you.